All right, hello. Today we're joined by Akanksha Yadav. Um, and she's currently a PhD student at CalTech. So I'll jump right into it. So, what are some common misconceptions that people have about your research interests, and how would you like to address them?
Um, yeah. Um, I I think I'll try to describe what my research looks like at the moment and give a sense of what I personally thought when I entered the field and sort of what I feel are like common misconceptions. Um, so I was coming from, I would say, a more engineering perspective, as being a BTech um, at a, I don't know, at a technological institute. We all have this like notion of fields like physics, chemistry, maths being more theory driven, and that there are some fundamental principles that we rely on to kind of build more complex things. Um, and essentially, I am currently in the field of biology. And thinking back, I feel like the notion I had when I was thinking of biology as an undergrad was more as a subject that you would just mug up for an exam, and that there were no conceptual bases that felt exciting that you could hold on to and try to build more complex things on top of it. I felt everything was essentially um, an outlier in some sense, that you couldn't couple things together to form like a big whole out of something more fundamental. And I think so. I wouldn't say that notion is entirely wrong, but I think so that there's some sort of beauty to it, in a way that the diversity in biology is so huge that it can feel like a challenge when you first see it. Everything seems to be an exception of its own, and that it seems like a problem that seems very hard to tackle. But I feel that it doesn't escape the realm of biology, like the realm of physics or chemistry. Like the fundamental principles are still very much what governs all of life. So I feel it's more of a matter of trying to um, do it both ways, as in trying to build simple things and to match the complexity, or try to like simplify the more complex things into simpler ways, and build principles based on that. So I don't think that was a correct way to approach the problem. I think so. The notion is, in a way, justified. The way people talk of biology in general, it feels very complex. So I think so. Yeah, coming into the field, I felt uh, more and more convinced that it's not a problem that one cannot tackle if thought about carefully, and especially with more advanced machine learning tools and AI coming, I feel more complex things can become more human approachable. So that's also super promising, at least in the recent years.
So
Yeah.
I think you mentioned some very interesting viewpoints towards the beginning of your response, which was that when you're looking at biology, it's there are a lot of things going on in there. Like there's the molecular aspect of it, like a molecular study which you do in biochemistry and whatnot, and you sort of analyze individual molecules and how they react within the uh, with other organic compounds and whatnot. But then there is the aspect of soft condensed matter physics where you have emergent properties um, coming out of those organic molecules, maybe polymerizing together or building up higher dimensional, well, like more complex structures. If, like, let's say if we could think of DNA being formed out of like just sequence of um, uh, proteins, right? Is
Nucleotides.
that's what they are?
Um,
Oh
yeah.
yes, sorry, nucleotides. Yeah, I'm really have to rough up on my biology. So yeah, so nucleotides, right?
Yes.
And so yeah, so like similar structures giving rise to more complex entities, and those complex entities have like a completely different set of properties to them than
Yeah.
they did at the earlier. So I think that's very interesting to note in biology, that you're also coming at it from a perspective of a reductionist approach, where you're constantly trying to simplify things to and like grind them down to their bits and pieces, whatever they might be made up of. But at the same
Yeah.
time, you're also sort of analyzing the emergent structure, and with properties which are completely different from the base ingredients. So I think
Yeah.
that's really interesting that you mentioned that. Um, so the next, would you like to add anything on that, or like, I mean, of course, I might have said some horrendously wrong things. Like, feel free to correct me, because I'm not really an expert, but by any shape or form in this field, and that's being diplomatic.
Uh, no, I think so. That's a very yeah. I think so. The one of the very I mean, it can be thought of as a like it's an emergent property problem, like at multiple scales
Yeah,
of length and time,
yeah,
because you bridge across, I don't know, like orders of magnitude of length and time scale in a single living organisms. And
yeah,
if you look at, yeah, the size of, I don't know, mammals, and the length scale of a molecule, and bridging all that, like it's almost and I think so at any level, I think so it's at any level an emergent property is a different set of problem of its own. It can be looked at lenses like from many different uh, levels. I think that makes it also like I mean, traditionally, I think people also in the field are kind of segregated in that sense that
uh,
there are very few people who look uh, at uh, at a series of emergent properties, if I can put it that
mm,
way, of biology. They, if just because it's like painstaking to do something with very carefully at any of these levels, it's like people are married to either one time scale or one length scale. If someone is looking at one protein, then they forget the 20,000 other protein coding genes, or the 3 billion other base pairs that nobody even knows what the rest of the genome is doing. So I feel like in trying to like focus on one aspect of that emergent behavior, it's like uh, people forget that it's even connected at so many scales.
yeah,
Yeah, it's easy to lose context, I think.
yeah.
Yeah.
All right. Okay. Um, okay, so I have a silly question, since this
Yeah.
podcast is actually about making things accessible. So you said nucleotides. So what is the difference between nucleotides and proteins? So like the ATCG thing, is that, are those called nucleotides?
Yeah, yeah. So the ATGC are called nucleotides, and the that make up DNA polymer or RNA polymer if it is a UGC, so T
Yeah.
instead of U, and they are kind of sugar molecules, kind of, well, modified sugar molecules, I would say, that form like hydrogen bonds between them for base pairing essentially, and that what makes like DNA or DNA double stranded, for example.
Yeah.
Yeah, and proteins are different kind of polymer because they're made up of amino acids, which are
Mm.
20 amino acids, and they're bonded by amide bonds, like they are single polymer chain, and everything has orientation, so it's a directed chain sort of, cannot be
Yeah.
the
Yeah.
order
Yeah.
matters basically.
Yeah.
So,
Yeah. Makes sense.
yeah.
Yeah. By the way, I just like to correct myself. When I said silly, I just meant that what people would consider to be silly, myself included. There are no silly questions. Um,
No, certainly not. No.
yeah. So moving on to the next question is, can you share like a pivotal experience that solidified your commitment to mastering your research interests?
Yeah. Um, I I think so. I mean, I am a major in chemistry, and going back, I think so. I really liked physical chemistry the most. It was very, um, I think so, understanding heavy in my in some crude way. I felt that it was principled, that there was. I don't know. I really like thermodynamics and kinetics and stat mech and yeah. So like ensemble properties, thinking about molecules, groups of molecules, and their behaviors essentially. So all of that was exciting. So I think so in my third year, I was really debating um, between kind of two options. In my second year, I was for the very first time, really excited about molecular biology. From doing a course at IIT Bombay under Professor Swati Patankar. Um, I think so for the first time I could see how biology was something that you could build. And as an engineer, that's very tantalizing that somebody tells you that you can build something from your own hands and that looks something like life. That was really exciting. Um, yeah, I in my third year, I think so I was debating between trying to go more into biology or go do some quantum chemistry. Really, like I really wanted also like the computational aspects of physical chemistry. So I ended up choosing biology. I think it felt um, something more testable in my mind that that you could uh, analyze it, but also go and do some experiments to verify it, and felt like a closed loop and satisfying in some sense. At least my idea of it at that point. Um, so yeah, I think so. Then I chose to spend more time trying to understand the research in biology better. Yeah, but that was kind of crystallizing that okay, I need to know more in biology, I think.
Right. So I'd just like to introduce a point here, and which is very interesting and ties to what we just discussed before this, which was when we were discussing this off the podcast, and I think this is really important because it ties very closely to the point that we just discussed, like the question that we just discussed, is that
Yeah.
when I'd asked you off the record, like off the podcast,
Yeah.
what kind of a pivotal experience that you had that really inspired you to do this? You said that being able to dissect life and reduce it into its parts really excited you, but the answer you gave right now was building up parts to get, like which results in life, which is extremely interesting because again it goes back to the multi-scale problem of how emergent properties look at different scale, right? One is the
Yeah.
reductionist approach where you're just going backwards, you're working your way to like you're trying to grind it to molecules and atoms and whatnot, and the other one is you're trying to see how complex phenomena arise out of simpler entities. By the way, that also reminds me, I had a conversation with a friend of mine long ago that so we were pondering over this question: how do amino acids know how to link up with each other in order to form proteins?
Yeah.
Like how does that happen? So that's just something that just popped into my mind because you'd use this phrase, how do simpler entities give rise to life by linking up together. So
Yeah.
yeah, but like any thoughts on that?
Yeah, I think so. Like, I mean, I know I gave like two different answers in two different directions, which almost point to like different kind of biologists in the field. Um, the reductionist one don't usually sound like the emergent ones, for example. Um, And the thing is that, like, I don't personally, I am not married to either direction. I feel like
Yeah.
the way you understand something as complex as biology is going both ways and
Yes.
trying to switch between those perspectives as need arises. I think that's, in my head, kind of an optimal approach. Certain simplifying principles are very powerful when you need to boil down to certain things. And on the other hand, complex things have their own subtleties that you cannot ignore,
Here,
uh,
here.
that are not fully, captured in simple things.
Yeah.
So it goes both ways, I think,
Yeah.
so in my head.
Absolutely.
But going back to the question that, amino acids and everything that is biochemistry, like if you put it that way, like when you ascribe a function or a role to a molecule, it's like almost saying that a molecule has free will. Does it
Yeah.
have like um, does it have actually like a choice to do whatever it wants, or is it like
Exactly.
pre-programmed to like do this exact thing? And I think
Yeah.
so it's more of the um, second kind. I think so. It's not um, like at the level of the molecules. It's not as if the protein knows that it has to be at XYZ position at time, whatever. Um, I think so. The system is such created that um, it is like we think of biology as an out of equilibrium condition. So essentially, you need to maintain energy gradients and ionic gradients. Essentially, nothing should be balanced across what is contained inside like a membrane that's called life, and everything that's outside is water. So to be able to sustain that like life thing, you need things not in balance. That there should be a flow of um, molecules that will support, will give you energy, that will maintain your ionic balance. Um, and I think so. You can think of proteins also acting in a similar way, that there's always a forward rate and a reverse rate to things. In certain ideal situations, the forward rate is fast, so it goes on to do that thing. And then when it's done, it's switched back to the original state. So it's kind of like, and I think so that is one of the reason why like creating artificial life is not easy. Like you have to make somehow the perfect imbalance system to like kickstart something that has been alive for, I don't know, ages. Um, yeah.
So that, yeah, please.
No, but yeah, I think so. That's like, I don't think so. Molecule has free will, and it's kind of a programmed way to think of, even at the level of proteins. Like we think of them as like, I mean, I want to call it Maxwell demons, like Maxwell's demons, that they, which says that it's not free will, right? Like that they maintain, that they can toss around molecules from one side to another, and they can change conformations and
Yeah.
kind of do that for
Yeah.
yeah.
So I've got. Thank you so much for bringing that up. That's amazing because one way to think about life is the Maxwell's demon hypothesis that, okay, you're creating order out of chaos, and there's this Maxwell's demon sitting at the edge of the membrane who's not letting order collapse into disorder, and is somehow not letting entropy increase. So that's one point of view, which is right, and the other point of view is, and so wait, sorry, did you say that the Maxwell's demon theory, according to you, points towards free will, or is it the other way around for you personally?
I think so doesn't point to free will. I think so Maxwell's demons is kind of like a pre-programmed thing that's designed to behave in a certain way, and it just does that all the time. And yeah, I think so it's more of like, if it was, I mean, it can be a little bit confusing because
Yeah.
it's I mean, there are a lot of feedback loops, right? Like, which makes it slightly confusing. So, the ratchet is not forever on. Like, it's once it's turned on, it's not forever on. Like, there are certain conditions in which it will be off when some trigger happens, which are like the feedback loops inside the cell. So, but you can think of the feedback loops also as programmed things that are pre-programmed to happen when this ratchet has done X and created something. So I tend to think it's like if you can think of like well coordinated Maxwell demons, like many of them, in some feedback loop.
All right.
um,
Okay. So so I've got to. Sorry. Did I cut you off? Please go on.
no.
All right.
Go on.
So I've got another theory, which is that the feedback loops are set up in such a way that the entropy of the apparently ordered system is actually ever increasing. So no matter even if life from the outside looks like an ordered system, it's actually creating more disorder. So the entropy is ever increasing, and you're looking at it from like a keyhole perspective, and you're not looking at the other disorder that's arising out of those ordered structures. So in that case, if I were to take those two views as the only two that exist, for me,
Yeah.
if you had multiple Maxwell's demons like sitting inside your body on all the membranes and whatnot,
Yeah.
I'd say that those Maxwell's demons would actually like point towards free will, because there's something that's stopping the entropy from increasing, and the only other explanation for the entropy to keep increasing is that it's just like a different perspective in which you've not considered the disorder that's been created by the ordered structure, which is the living being itself. What do you think?
Yeah. I mean, yeah, I think so. I find it like hard to like attach free will to, I guess, molecules also because um, I feel it has to do something like, which actually, I mean, could be the case. So the issue I have is that the way we study like any set of proteins or, for example, iron transporting or yeah, like ATP synthase, which generates ATP or energy. Um, like we always consider them in this context where it is sitting here, like on the membrane, doing this thing and exposed to very physiologic conditions. So uh, it's not removed out of context ever, right? Like I'm not saying an ATP synthase in some like what if in a normal cell suddenly there are 10,000 of them instead of 1,000. Like that is something abnormal, and then its behavior is kind of goes a little unpredictable because on an ensemble level something different could happen, or maybe I don't know. Then it will adjust to it, or what will happen to it. Like that becomes a little bit like its original function might not be the same as
Yeah,
what I thought before. So then I don't know how to say where it goes in the free will
yeah, yeah, yeah,
um,
yeah,
axis.
yeah. So I think so. The one flaw in my particular hypothesis or analysis that I just gave right now is probably it's too reductive, and it doesn't like, again, it's like looking at it through a keyhole and not like for example, the example that you just took probably repudiates the analysis that I just gave. So I think yeah, I'm not qualified to talk about this.
No, it's yeah. I mean, it's yeah. I have also not done super intensive analysis on it. Just a few thoughts here and there.
But it's really interesting because, like, if you combine philosophy with the second law of thermodynamics and then you apply it to biology, you get some really interesting trains of thoughts like we just discussed. I think that's something
Yeah.
that we can explore, if you're game in the future for another podcast episode. Yeah. So moving on. Uh, how would you describe your approach to problem solving within your research interests, considering the unique challenges it presents?
Um, yeah, I think so. Yeah, I think so. It goes back to um, coming from a little bit of a engineering background and having um, some sort of like I don't know, I want to call it core principles, like personal core principles as a scientist, to be more, to have to do an active effort towards being as quantitative as I can about the way I approach things. Um, I think so. In that sense, I um, when I started out like doing research in biology, kind of the first field that I was working on was more computational biology, looking at like large data sets of, for example, the sequence of DNA inside a human cell and comparing it with all the RNA that is present inside a human cell, and then comparing it in like different tissues in the heart, in the skin, in the muscles, in the brain, and what that profile looks like. So there you deal with just big matrices of numbers. Um, so I think so from like, and that was satisfying, I would say, in a way that uh, you could make very quantitative statements about correlations and covariance, and yeah, you could see like if you do a PCA on thousands of individuals or sequence across Europe, you can actually like just across plotting across PC1 and PC2, you can actually see ancestry. Like you could see the Swedes being different from English people, and I don't know, like that's certainly cool. Like that there are signatures in your genome that co-vary, and that's enough signature to tell apart that where you're from, or uh, that you do carry like genes that are, I don't know, local to the place, something of that sort. But yeah, those kind of quantitative analysis are turned out to be pretty rare when I kind of had a switch over to doing more wet lab biology experiments, just because the kind of data you collect is not that rich in the first place, and it's usually low n. So you cannot low n, meaning that you don't have as many data points to do very statistically significant kind of analysis. If you have just 3 data points, then you cannot make very qualitative statements about things. So, yeah, I think so. My approach is still trying to find ways to have more data and more kinds of data in biology. I feel very excited about the opportunities there. Personally, I am also quite, I think I've spent quite some time trying to think about molecular tools that actually um, allow you to collect such larger data sets, I think, in different kinds of biology fields. So, yeah, I think so. My approach is still um, trying to collect bigger data sets, more comprehensive data sets, and um, trying to analyze them using all the statistical tools we have.
Yeah, yeah. So, so I'd like to address 2 points. One of the things that you said was molecular tools to collect more data sets. Could you like expand on that?
Yes. So I think so. Maybe I can talk a bit about this couple decades of research in biology if I had to give like an overview. Um, I think so. Something revolutionary that has happened is that the cost of um, um, sequencing DNA has gone down like orders of magnitude over the last few decades, which means that we can now sequence DNA pretty cheaply of like billions of base pairs in a few days. And just to give a kind of perspective, in 2000 or 2001, the Human Genome Project was kind of established, where they spent like billions of dollars to sequence the first human genome of 3 billion base pairs. And at that point, it was, as you can see, like it was really expensive to sequence DNA. So people since then have tried to like parallelize the sequencing platform, build like automated tools to make it easy. But yeah, long story short, I think so now the thing is that any kind of molecular information that you could encode in DNA is super sequenceable and very cheaply. So now people are trying to harness it to do, like, for example, well, you can sequence DNA for sure. You can sequence RNA as well by converting it into DNA, which can be done by enzymes. So essentially, you have DNA and RNA, which is kind of one third of central dogma, kind of sequenced. So you know inside a cell, it's genomic sequence, it's RNA contents, and yeah, essentially anything you can tag with DNA, you could sequence. So those are kind of molecular info. Yeah.
So one of the things that I've never gotten about sequencing is, why is sequencing done? Like, what's the, you know, what's, I mean, obviously there's some use to it, but what's the use of sequencing the human genome? Like, what kind of inferences or conclusions can you get out of that analysis?
So I think so. Yeah, one thing to understand is that every cell inside an entire living organism has a nucleus, which has a DNA sequence, which is shared across all your cells. So you could think of DNA as a blueprint for you. That is your blueprint that played out like a recorder and gave rise to you as a human being, you as a bacteria, you as any living thing, actually. So the I think so. Sequencing DNA was very tantalizing in the beginning, thinking that, oh, uh, what if you just sequence this kind of, tape, this blueprint, and once you do that, maybe you'd be able to understand how an organism plays out of it, because you would see the whole sequence, and you could compare individuals and try to see which parts are conserved. It would all make sense that, oh, these are the key things that are important because they're conserved across humans or across bacteria. And we would, I mean, it was kind of wishful thinking to think that we would suddenly make sense out of just 80 GCs in a
Yeah.
sequence, all of a sudden, just sequencing it.
And
But
every
that,
kind of disease would be solved, cancer, autoimmune, everything would just be solved by inferring.
yeah, yeah, I mean, yeah, I think so. That was the hope. I mean, to some extent, yeah, something like you could do genome-wide association analysis where you look at maybe thousands of people having disease, not having disease, and doing correlations across these different groups, and suddenly things would pop out. Oh, breast cancer mutation only in these batch of people, and then you could go and correct it or like target it. I think so. People had the hope that everything would be like that, and
Yeah,
the things turned out that most actually diseases are not monogenic, meaning that it was not caused by one single mutation. There are a
yeah,
bunch of, yeah, like
yeah,
differences in the genome that cause
yeah.
either, yeah.
So, if I understand correctly, a mutation is basically any um, difference. I mean, it's a mutation in something, right? So it's a mutation
Right.
in the sequencing of your DNA, right?
Exactly. Yeah,
Yeah.
yeah, yeah. But I mean, mutation you could is always with reference to something.
Yeah.
So it
Exactly.
means
Yeah.
that like in most humans at position 280 there's a C, but in patients who have like this very niche kind of breast cancer, it's a T or something. So and that explains why they have a mutation in the protein that is coded by their gene, and then you can say for sure, okay, then it relates to breast cancer because that protein is super important in regulating something.
Yeah. All right. Okay. And so the most common mutation I've heard about is the BRCA2 gene mutation, right?
Yes, which is the breast cancer.
Yeah. And that's
Yes.
the same as what you talked about, or the particular position, the 208th
Yeah,
position
I
or
don't
something.
know. Well, 280 was just random, but the BRCA gene or mutation is one of the most, I guess, one of the most successful example cases where sequencing gave rise to an understanding of that position that was dominant in breast cancer, female breast cancer cases. So, yeah.
That's amazing. Thank you for that analysis.
Yeah, but I actually need to elaborate a bit more.
Yeah,
So
please.
yeah, if you think of DNA as blueprint, the idea was okay. Once you sequence it, maybe you'll understand something about uh, what gives rise to, for example, diseases, but also the fact that um, you can analyze which part of the genome is codes for proteins. And once you know the sequence, the differences would also explain the difference in different kinds of proteins, maybe like which
Yeah.
mutant proteins are made, if mutants are there in the protein coding region. So the central dogma basically says that, well, it says, gives this idea that you have DNA, it gets transcribed, or information passes from DNA to RNA to protein. Um, in like DNA gets transcribed to RNA. RNA is this intermediate molecule, and RNA gets translated to proteins, and proteins are basically uh, kind of the players that are in the cytoplasm and do things for you to basically, yeah, sustain life. So that was the idea that something at the protein level could be identified that was different between individuals and could explain differences.
So at the end of the day, even after doing all of that analysis, even though
Mhm.
because we already discussed the role of multiple such mutations actually giving rise to uh, diseases of a particular sort,
Yes.
it's just because the BRCA2 gene mutation has a very strong correlation with the occurrence of such diseases is because it's recognized as a like a pseudo causal,
Yeah.
mutation, but
Yes.
it's not. We don't actually know if it's actually what's causing it. Like,
Uh,
is it? Yeah,
yeah. I mean, yeah. I'm not sure about the. Um, I want to say I think so. People know that it's causal, but I have to double check. Um, yeah, it's that if you correct it, maybe you. I think so. People do want to do that, and I think so. Then that's the reason that if
yeah,
it's causal, then if you correct it, you shouldn't. Like if you correct it at birth, for example, then you shouldn't get that disease or shouldn't have that
yeah.
predisposition. Um, in that case, I want to say it's causal actually. And monogenic meaning, I think so. Also that I think so. That's yeah,
Like
if
just the
you have, if
BRCA2.
you
Yeah.
know, yeah, if you have that, then it's like sure that you will get the disease,
All right, all
something of
right,
that
all
sort.
right,
Yeah.
all right.
In general, direction of causality is difficult to establish.
Yeah,
I think so.
yeah.
Yeah. Yeah.
So this actually brings me back to one of the things that we discussed. It, with respect to the problem-solving approach, you said that there are so many variables to consider, And because it's the fact that doing experiments with biological entities has to be carried out in a statistical ensemble. Like, you can't really carry out the analysis over a single molecule because you don't have life in a single molecule. So because you have so many layers of, like, a multi like a 4D chess kind of thing, you don't really know which move affected the final game.
Yeah, yeah,
So
something of that sort.
yeah. So
Yeah, I think so that,
yeah, please.
yeah, makes,
Yeah, please go on.
yeah, I think so that also makes it hard to do, um, causal inference all the way. I want to say
Yeah,
it like that.
yeah,
Um, that it was not enough to just sequence the genome, like maybe, I don't know, there you can think of it as like maybe there are ways to compensate that mutation that we just don't know because we have not seen the whole system. And DNA is just the first entry point into it. So all those, like, there's essentially a lot of redundancies built into the system to keep you alive.
yeah.
So which makes it tricky.
And we don't know them all.
Yeah, you never know them all. I
We
think.
never know them all. Yeah. All right. So the next question is, how would you explain your current research interests to someone like me, like a 26-year-old with a STEM background, and juxtapose that and sort of boil it down for a 10th grader? And also, if like, what's the most exciting aspect of your research that you'd like to convey to the audience, myself included, obviously?
Yeah, as I have to say, I really love my lab. I work in the Shapiro lab here at CalTech, and we are essentially a huge team of scientists who want to build ultrasound-based technologies um, to kind of probe deeper into more complex organisms. So I'll try to break that into simpler terms. So I think so. Most typical ways of trying to understand biological phenomena is literally by seeing them happen in real time. That's the most, I would argue, it's the most information dense way of actually studying biology. And historically, I think so, people have spent a lot of time trying to use light to look at things moving inside cells, inside animals, inside bacteria, inside any living thing, really, um, to try to understand what is happening. And the way they would do this is, for example, they would it's a jargon that I'm going to use. You can tag anything. Tagging meaning that, let's say you have a fluorescent molecule. Fluorescent molecule is a molecule that when you shine light on it, it will turn bright. I think that means you can see it under a microscope if it's large enough that you can see it with your naked eye. But essentially, you have cameras that you can use to capture all that light and see where it's coming from. It gives you like the spatial, and you record it along a time course that gives you the time information. So essentially, fluorescence is a great way to attach to any molecule of your interest in any organism. Just have a couple of cameras, pretty much like in your iPhone. Just capture it and just play it. Try to see patterns. Try to see where things are going. Uh, if you can have two colors, then see where X is going when Y is going there. Things like that you can tag different molecules and see what's happening. The thing is, light is great. Light is very bright. You can see light. The bad thing is when you start to look at organisms which are bigger in size and not typically transparent, such as opaque animals like us, or like what we study in the lab are typically rodent models such as mouse. Light doesn't penetrate deep into the tissue, that you could observe something, a signal that's coming, for example, from the gut or from deep inside the brain, like in the central nervous system. So you are limited to kind of like seeing things on the periphery at a very bad resolution. I would say something like an organ scale resolution. That means that you cannot ask questions about things that are happening at the cellular level. If there was a change that's happening in a cell, essentially you average it out across a big region of an organ, and then you kind of lose if there was one cell that was doing something very unique and misbehaving in a particular way that you wanted to chase it down. So I think so. Ultrasound is kind of lower frequency energy that you can harness to penetrate deeper inside tissue, because you can, scan your baby when your mommy is pregnant and see a baby growing inside a mother's womb, and that is essentially ultrasound. So it can penetrate very well deep inside opaque tissues. And we are essentially trying to harness that to image deep inside the tissue, but at a very fine resolution, almost at the cellular scale. So that is kind of the, I would say, our motivating principle to build technologies using ultrasound. My advisor was the kind of first one to actually find, we call them genetically encoded ultrasound contrast proteins, that he picked up from bacteria actually. So these are proteins that trap gas, and basically they're stable inside cells. And because they trap gas, you have like a water and gas interface that has differential
ultrasound
contrast for
signatures.
ultrasound. Yeah. So we basically harness these proteins that are originally from bacteria and try to express them in cells from mammals and put them inside, deep inside tissue, and try to image them from the outside without any issues, and without having to worry about. Yeah, that's something that we do.
So that's really interesting. So one of the things that came to my mind is, you said that with bigger organisms, which are not transparent, unlike microorganisms, you're unable to perform more involved analysis because you obviously can't see inside the organism, and you can't use light as a probe. So I think there was something that you're interested in with respect to probing the nervous system in mice, and like just having something which just captures all of the information, how it travels, where it travels, and how it's processed within the nervous system. Would you
Yes.
like to talk about that?
Yeah, sure. So I can actually tie it back to kind of my initial enthusiasm about building tools, as I was describing. One of the tools that we want to develop based on this ultrasound contrast protein is something that can sense calcium. So when I say sense calcium, it means that it has different acoustic properties or contrast properties in response to different concentrations of calcium. When there's high calcium, it has, let's say, high acoustic contrast, and when there's low calcium in the surroundings, it has low contrast. And what this allows us to do is basically, when you express them in cells such as neurons, which are essentially the basic unit of a brain. It allows us to sense calcium, and calcium signals are kind of a proxy for neuronal activity. So it is the first thing that gets kind of perturbed when an action potential is triggered. So it causes an influx of a lot of calcium as soon as you have an action potential firing. So essentially, sensing calcium is a way to say that, or even calcium traces along time is kind of a proxy for the neuronal activity, and neuronal activity tracking is kind of the, I would say, one of the fundamental ways in which we think a neuron actually computes. So it is something that almost is like a, I don't know, dream for every neuroscientist to be able to know what processing is happening at the level of each neuron across an entire brain. Yeah, in mouse, it is typically challenging just because you don't have access or a way to actually record from each neuron. Yeah, especially across the entire volume. So, yeah, that's
So
the
so
goal.
so I think I'd just like to point out for the audience that neural circuits, neural networks within living organisms, mammalian bodies especially, are not. I mean, we used to have this misconception, I think, in school that neural networks are just like electrical circuits, but they're vastly different, and you can't actually use electrical circuits to model neural networks, and they fail really badly because there have been several studies which have found that action potentials, the way that action potentials work in triggering other neurons, in the network, is extremely different from uh, electrical circuits, and the functioning cannot be explained by a simple analogy. And there's something much more involved going on. And yeah, would you like to weigh in on that?
Yeah, sure. Yeah, I think so. Even at the level of, I think so, single um, neuron, um, I think so. I personally feel it's an oversimplification to even say it's similar to what a neuron means in a neuronal network. Like
Yeah,
it's not just a bunch of inputs coming in that gets added up in some uniform, well, not uniform, but not the same nonlinear bias is applied to each neuron in some layer, and then it activates the next set of neurons, and that remains the same, I guess. Uh, I think so. The computation, even at the level of a single neuron, is not um, well understood, I would say, because yeah, I mean it does receive inputs, and you can think of it as a input output kind of a function generator, um, but there is not like I think so. There's some even, like for example, if you activate the upstream neuron and are looking at what happens at the downstream neuron, I think so even that process is kind of a statistical process that
yeah,
every time you do it, it's not that the same dendrite and it
yeah,
gives like the same weight to the neuron that you can add up the same way.
yeah,
Sometimes it's equally shared between dendrites. Sometimes one weighs more than the other. Depends on the geometry. Depends on some other local input from something that's inhibitory, and it kind of like
yeah.
shunts that input at some point of time. So it's yeah, I would say the neurons are densely connected, and addition, multiplication, subtraction, I don't know what all computation can happen even at very local synapse level and can change over time. So yeah, I think so it's more involved and needs more um, experiments, I think, to
Yeah,
be able to
yeah.
boil them down into computations.
Very far away from determinism, basically. Can't
Yeah.
use.
Yeah.
So there was something with respect to some of your research interests that I was interested in, because we talked about this before, which was one of them was protein engineering and the usage of models like AlphaFold in the same. Would you like to take up like how data generation can help in protein engineering?
Yeah. Um, yeah, I think so. I would just go back to like giving a little bit of a background about kind of the AlphaFold moment. I think so. Uh, I mean, I mean, certainly appreciate AlphaFold as a revolutionary model, but somehow they kind of don't look carefully at the fact that AlphaFold relied super heavily on a very well curated database that has been there for um, at least 20 years now. I think some more. I think so. It started in the maybe in the 1980s or 90s. And I just recently learned that PDB was so like PDB is the database that has this essentially all XYZ coordinates of solved crystal structures, which means that you know what a protein looks like in three dimensions by either using I don't know, cryo-EM or essentially using X-rays or high energy rays to kind of find out where all the atoms are in the protein molecule. So essentially, PDB is a database that stores this information for many different kinds of proteins, and it is curated in a way that's very standardized. That means each entry will look very similar. It's the formats for storing the coordinates is very standardized, and it was standardized to my very surprise, and it was stored in electronic format from its like conception. I mean, whenever they thought that they should make a PDB for doing this thing, they made it public, they made it standardized, they made it electronic, uh, which to me is like super, uh, like it's amazing foresight. I think to have this notion that this is something if we had a platform for sharing, it could be meaningful for people researchers to have access to this and then every like and this is separate to any of the publishing like realm. This is open to this is meant for sharing. This is meant for putting your science out there. Is meant for helping others to be easily accessible to such information. So I feel that's also not so common as an effort in biology in general. So yeah, that the idea was there. This platform existed. So basically every person, every researcher, every group that solved crystal structures would then submit their like structures to this database in the same standardized format. And over time, like you can look at graphs, the number of structures has been increasing, and to the extent that now it makes sense to actually do good inference on it. And I think so. When AlphaFold came in, I think so. It was CASP 14, and CASP also should get. CASP is essentially kind of a protein blind protein structure prediction challenge that people developed to kind of benchmark these computational tools that could use structures that were, for example, in that particular year not put into the database yet, that were recently solved, that if you develop models, you could test them on this blind kind of data set that the model hasn't seen, and get scores for how well it does with respect to actual structure. So this was also a competition that people hosted as a public thing that due to benchmark these models continuously. I think so that's also a really commendable effort. And then Google's DeepMind came in as a team that participated in this, and they then published AlphaFold as a model that in that year actually surpassed any other kind of model that people were kind of using before. And I think so that's huge because it got accuracy scores which was pretty unlike any other model that one had ever seen. Yeah, I think so. Since then, you could say that they expanded really quickly, and now we almost don't even like as a first pass. I think so. It's a really useful tool to think about if you have a given protein sequence and you want to know its structure, to even think about it, to have a 3D molecule in your mind. You just input it and get an output 3D.
Yeah.
I think so. That's where it is really helpful. I think so. To the extent that how its impact would spread across, different fields in biology, I personally am waiting to see results of it that are more convincing. And I think so. My perspective there being that as a protein engineer, I have spent quite some time trying to, develop better versions of proteins by manually changing individual amino acids and trying to screen for certain properties that I care about. And that is challenging. It's usually, we used to say this in our lab, that it's usually easy to break a protein than make one. Yeah, go ahead.
So I just like to state something for the audience because there are some words I think they probably won't be able to recognize. So what Akanksha has been referring to here is with respect to AlphaFold. So it's basically these proteins. They're like really long polymers of amino acids, right? And they fold onto each other, and how they fold onto each other determines how they interact with their surrounding within the tissue. By the way, please feel free to correct me if I go wrong. Yeah, just like stop me if I'm mistaken at any point in time. And the reason that the structure is so important is because you have a lot of reaction kinetics as well that gets dictated by the structure of the proteins. So if you know what the protein is made out of, and if you have a tool that can give you the structure that it will fold into, that will be a huge breakthrough, because then you'll be able to determine, well, not determine, but reasonably extrapolate how a particular protein would react with its surroundings for a particular biochemical reaction. So, and that's why it's really an intriguing topic to be explored in biochemistry.
Yeah, I think so. I would want to add on to that I think so. It's still, when I think of like a 3D structure of a protein, I feel, I mean, yeah, just to give a picture of, inside cells, just as everything in even in water, no molecule is like actually stable in just one state. These are bonds that vibrate. They interact with water. That means like the entire protein is kind of a jiggly mess of like atoms, you could think of. And in a protein structure, essentially what you're seeing is kind of if you can think of this jiggly mess of thing dancing around, kind of the 3D structure that we see in PDB is more like the most stable structure of its entire ensemble. If you could find different states that it could live in, It might be the most long-lived state, for example. So it is like just one n equal to 1 kind of sample of these lot of samples that you are not seeing when you think of this stable structure kind of picture. Um, and I feel that there are very few methods that actually give you a sense of the kind of true kinetics of proteins. And some of like I spent some time doing NMR on proteins, which kind of samples some of these short-lived states, and doing some level of computation as well on some sampled states from cryo-EM. So cryo-EM has this like you flash freeze bunch of proteins and capture them in like different kind of orientations, and then you kind of average them across all these states. So you still have some averaging, but can still sample some variety of states. And then you try to infer like moving from one state to another using some kind of physics informed modeling approach. So I feel like it's still in essence, if you could, one would argue that you never really truly see the protein and all of its conformations ever in any technique in a more biological relevant, I don't know, environment.
Yeah.
You only see samples of it. So yeah, I think so. Like personally, if you like, you can think of structure as kind of one sample of that protein. It can be like I think so. People are still kind of exploring like how much physics-based modeling can inform us about its dynamics and where it can lead us. How I don't know generative modeling can be helpful in that case. Typically, I think so. Most impactful information from some of these stable states is one where you bind something actually, like when you can bind small molecule and stabilize it. I think so it directly translates into I don't know, therapeutic spaces where you have small molecules stabilizing proteins, or um, locking the protein into certain conformations, not letting it do something like that. So
Yep.
in that sense, I feel it's kind of more impactful.
Yeah. So I just like to add like a note to that is that even though you know we're doing all of these statistical averaging, even that helps because if you're able to predict like the most ubiquitous um, state that the particular protein exists in, the structure that it exists in, you'll still be able to extrapolate a lot of things just on the basis of that. And
Yeah.
and like biology is inherently it's all about complex systems. So it's not about it's actually going from like one scale of complexity to the other that's the problem. It's because every time you're trying to predict something, you're crossing a scale of an order of magnitude of some kind of complexity. So you're jumping from a particular just for example with AlphaFold, it's just you give the chemical composition of the protein and you're trying to predict the structure, which is itself and a sort of a very difficult task to do.
Yeah.
And yeah, and that forms like a major foundation for how chemical reactions progress with respect to the kinetics. Um, so would you be able to shine some light upon binder proteins and stuff like that? Because I think that relates closely with the structure, or I may be wrong. Like, I have no clue. I'm just like,
Uh, I mean, yeah, I mean, I wouldn't like binder proteins as in um, well, I mean, I think of proteins as things that bind to other things.
Yeah.
That's kind of ubiquitous thing that they have to do to kind of transmit information, right? Like it has either it has to bind on another protein to like uh, be a part of a signaling cascade, or it has to bind another molecule, not necessarily a protein always. Could be RNA, it could be a small molecule, it could be an ion, it
My
could
apologies.
be
My
yeah.
apologies.
I mean,
It's actually no, no, no. I apologize. It's not binder proteins. It's binding proteins. There are proteins that bind two or more molecules together.
oh, I see.
Yeah, yeah. My
Um,
apologies. And they are important in regulating the transport, metabolism, and bioavailability of other molecules such as drugs, vitamins, fatty acids, and steroid hormones. And so
yeah.
the reason I brought this up is because the shape influences how they bind these, like bind separate molecules together, right? I might.
Yeah, yeah, I think so. I mean, yeah, I so for example, if you think of them as well, I mean, as a protein structure, um, then the things that they bind to, for example, especially in all these externally, so all these, I would say, like signaling proteins, they are usually because their function is to actually send something on the from the outside of the cell and transmit that information to the inside. That means that they have to send something like some ion, I don't know, calcium, magnesium, or some molecule like glucose, some neuropeptide, which is a small peptide, from the outside and change its conformation and then do something inside. And usually that means that the thing that they're attaching to is actually not a protein, right? Like when at least in AlphaFold, when it was all protein based, there was no a priori presence of anything on the outside for it to like there was no reason for it to sense something, you know.
Yeah,
In the model itself, it was
yeah,
just proteins in a chain,
yeah,
and that was it. So I think so. I mean, the tantalizing feature is that I mean, people when it came out were debating: did it actually learn protein folding? As in, like, you give it a sequence, and does it actually know the physics of folding that gives it a structure? And that is what it learned. That would be amazing. But I think so. More evidence points to the fact that because it uses a multiple sequence alignment in its like input, which means that it can align similar kind of sequences in the sequence space, like of the amino acids. It can basically try to find positions in the protein sequence that co-occur more often than random, and try to kind of position them such that they are close by. So it is more of like trying to preserve the 3D structure of the protein family than actually trying to learn de novo protein folding, I think.
yes,
And
yes.
that's the current understanding, I think, of how it works
Yeah, yeah.
internally.
So it's not like a universal type of thing.
Yeah.
It's just that it's learned um, within a very specific domain. It's learned how those proteins are arranged in like a 3D structure, and it's somehow learned to, I wouldn't say extrapolate, because extrapolation is you're taking outside the domain, but I think it's just that because of the data set might have been ubiquitous in the types of proteins it's trying to predict the structures of, it might have
Yeah.
just learned how to sort of just learn the learn to
Yeah, I think so.
interpolate rather
Yeah.
than extrapolate.
Yeah, I think so. Interpolate is
Is
probably the right. Yeah, it's more like you learn like local folding motifs or something,
yeah,
and just patch them together to form something meaningful.
yeah,
Is how I see it.
yeah,
Yeah, more of that kind of thing,
yeah,
and that's
and
why it's
please.
I think so. That's why it could be a little, uh, I don't know. I mean, I have to look at, I've seen like both kind of papers saying how useful it is for actually like single mutations, like single point, like single residue changes, because it's very local. You, you would think that because it doesn't care about that single position as long as the local motif is still the same, that it wouldn't predict very drastic structural changes caused by single point mutations, which make it uh, a little bit less useful. For example,
Yeah.
like you say, in if in the BRCA mutation thing, there's something at the protein level that's changing, but you cannot see that change at the protein level uh, using predictions, then it's a not so. I mean, that might be kind of a thing that needs more optimization, kind of thing.
Yeah. Yeah.
Yeah.
Yeah. Maybe it's just by sheer happenstance that the 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 training data and what it has been able to predict didn't have any drastic structural changes on local genetic mutations. I think that's basically what you're trying to say, right? Just because that the spectrum of structural changes because of local changes had been continuous, it was able to interpolate. But like, let's say a single mutation somewhere led to like a drastic um, change in the entire structure. I think it would not have been able to predict that. How do you feel about that
Yeah,
argument?
yeah. I mean, I think so. It could be either that, um, or I mean, there's this other aspect as well. I mean, not every um, like point mutations could manifest as different kind of mechanisms being altered in cells. It's not always just that is what I was kind of hinting to, that it's not always a big structural change
Hmm.
that might be the reason.
Yeah.
Um, for example, I can give you one example. Like you wouldn't, I don't know. It was uh, I mean, a priori, it feels surprising. For example, if you changed uh, a protein's localization inside cell by changing a single residue, like nothing about its structure changed too much, but somehow now it's not being like it was supposed to go to the plasma membrane to be able to act as a good receptor to things, but now it's getting stuck in the inner membranes, for example.
Hmm.
So it's not there on the outside where it has to sense. Now it cannot sense
Yep.
something.
Yeah.
So those kind of effects are not
Difficult.
always obviously structural, or
Yeah.
those are more mechanistic and
Yeah.
yeah,
Yeah. 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 That's
yeah.
really interesting because no, that's actually very interesting. I hope you're I'm not boring you because I'm
No,
droning
not
this
at
particular
all.
topic. Uh,
No, not.
because this is if you look at whatever we just discussed, right? The structures,
Mhm.
the
Yeah.
data that AlphaFold would have been trained on, the model of AlphaFold, the PD B that
PDB,
you
yeah.
yeah the
Yeah.
PDB that you referred to. It's
Mhm.
possible that the data set contained the kind of complexity that would have been that could have been captured in just in one dimension in the same, like it contained complexities within the same. I'm trying to define like a hyperplane over which you're
Mhm.
sort of like defining complexity. Like you jump from
Yes.
one hyperplane to the other when the
Mhm.
complexity changes. So the
Yeah.
example that you gave, like let's say you just
Yeah.
changed the particular, in the example that you give, that it won't be able to cross the membrane. So that's
Right.
probably you're operating on a different hyperplane, and you won't be able to predict that from this particular hyperplane,
Yeah. Yeah.
of that complexity. So
Mhm.
I think it's because you're just looking at the structure. You're not actually looking at any property that sort of goes
It was.
outside. Yeah,
Yeah.
yeah,
Yeah. And it could be that like y- the structure could be telling you about, you know, not localizing, just that you're not asking that question,
Yeah,
sort of thing.
yeah,
Um,
yeah.
yeah.
But even if you did, it'd probably be incredibly hard. Like you'd need tons of data. You need tons of data, and you would need to do tons of mathematical transformations to that data to be able to have for the data to be interpretable for the program,
Um,
artificially intelligent.
yeah, I mean, I feel, but like,
Yeah,
I mean, I think, uh, I feel that, I mean, AI is powerful in ways that it's sometimes not intuitive.
yeah.
And I can probably, yeah, I'm trying to think. I mean, it is more specific, but I don't want to like go into the specifics because it would be hard to like lay it all down. But I feel, any kind of measurements that's crude, but on a is done at a large scale, that we as humans don't see information in, might still have some hidden information
Hmm.
of
Yeah.
properties that we are not seeing right, just because we don't have the capacity to. And it's just something that we should ask more. And I feel I saw this in a couple of like biological kind of data sets where um, I can give you an example. I think so, like in a crude way. So we there was a lab I knew. They were looking at very large scale, like cryo-EM images of um, uh, like of a Drosophila brain, for example. And if you look at cryo-EM images, like you can, if you have good eyes, you can tell where the membrane is, where the synapse is. You can tell out some structures, but um, you can absolutely not know any molecular information. You can see structures like that look like boundaries. It's all black and white, black and white. And if you're not a biologist, you can tell nothing. Yeah, they so in Drosophila, I think so. There's this notion that each synapse has it transmits one kind of like, let's say, neurotransmitter, like one kind of molecule, and they kind of like uh, from some known knowledge of which part of the brain it was, they could like manually annotated a few of those synapse like structures to a few molecules that they knew would be present here, just based on biological knowledge, and trained a model to be able to predict that across the entire brain. And they found that it was actually able to predict something from EM data. EM data is just images of boundaries,
Wow. Yeah.
where you cannot even, there is no molecule there. There is maybe shapes of something that has that molecule. There is no molecular information, I'm sure, because at EM resolution you cannot see molecules. But something about the shapes of maybe vesicles or the local structure that is very unique to a particular kind of molecule makes it recognizable somehow, that it is able to classify it for every synapse. And they actually went back and did experiments to check if what it predicted was real, and it was maybe for more than 80% of the synapses that was correct. And I was like stupefied because I mean, one wouldn't even think of that question in the first place. Like, there is no prior reason to even believe that, a image that doesn't even have the information per se of something should have any, signatures of maybe, yeah, indirect signatures of something so important. So yeah, I feel like if you have it in a image or any kind of like pseudo uh, even layers removed from it, I feel you can still ask those questions about, localization or some other Yeah, it's farly removed things even, as long as you have the data.
So I was not expecting that. Oh, all right.
Yeah.
So I think would you like to talk about your interests in CAR T cell therapy?
Oh, sure. Yeah. Um, yeah, I'm trying to like tie it back a bit to kind of yeah, all the engineering aspects of biology. Yeah. So let me think. Yeah, I would say CAR T cell therapy is sort of this kind of new way of thinking about how to harness immunology to our own needs. For the longest time, I think cancer has been kind of one of those kind of diseases where anything you try, you cannot pin it down to any one thing. Kind of makes therapy hard to be precise. It has a lot of side effects. Rate of success has been low. And yeah, as engineers, it's kind of like you would feel a little bit demotivated by something that happening for such a long time in the field. So I think so. The idea had been there, but it really seemed tricky and kind of risky to try it. But the basic premise for it is cancer cells are these kind of cells that have gained a superpower in your body by undergoing certain mutations to replicate out of control so that they just eat up nutrients from your body. They grow uncontrollably and over time kind of harm the body, but somehow are also super sneaky in a way that your own immune system that is usually capable at recognizing such abnormal activities happening inside the body is somehow not able to see these weird kind of cells doing this weird kind of behavior that is actually harmful for the body. And yeah, I think so. The other thing is that the cancer cells are actually your cells as opposed to a foreign body that, for example, when you have a bacterial inflammation, it's a bacteria that's a different kind of cell, which has very recognizably differences as compared to a mammalian cell. So the immune system is actually also a very analogous to a neural network in some sense. That it is a system that is capable of kind of recognizing foreign bodies, and it kind of trains its cells to become perfect at recognizing this single foreign body, and stores that in its memory as kind of specialized kind of cells that it has trained to recognize one particular kind of infection throughout your life actually, so that a second infection is immediately recognized and kind of dealt with as soon as it enters the body. So in a way, it is also like a training system, that training kind of algorithm that happens in your immune cell. So the idea with CAR T cell is that the cancer cells are tricky because they are your own cells, they are doing something finicky, and your body is not able to see them as something foreign because they are your own cells essentially. And the body will not want to reject your own cells. But the cancer cells do have some molecular signatures that are slightly different from normal cells in that they express certain different kind of proteins on their membranes. So the trick is to kind of extract your own immune cells, like from draw blood basically from the human being, and insert kind of new genes that are very potent. These can be engineered again. What you introduce into the CAR T cell is a own engineering problem of its own. What you can choose really anything that is super efficient at being able to recognize the cancer cells better than your normal cells. So you can design new molecules, design new receptors, design new kind of like binding things that you can introduce into these immune cells and re-inject them back into the human. So now, and immune cells are kind of your basically in your blood, so they do travel all across your body and have access to cancer cells, normal cells, everywhere. So the goal is that you just need to initiate an initial recognition event so that your immune system is alerted to the presence of something harmful. And just that trigger is enough to like lead into this cascade of your own immune system then causing an immune reaction against the tumor to kind of suppress it and kill those cells that it now recognizes as cancerous. So yeah, that kind of like is like creating a
Yeah.
catalyst for your immune system. So
Yeah.
that has been really transformative, and yeah,
So,
it's
so,
being pushed more.
so you're basically taking live T cells from the patient blood, and then
Yeah.
in an artificial environment, training them to fight certain types of things which would otherwise cause the cancer, and then
Mhm.
and then because now you have this genetically modified, um, like it's replicated so many times, and certain kinds of genetic mutations, like survival of the fittest kind of thing. Out of those, the genetically modified T cells have survived, and then you put them back. Is it, or maybe I'm not entirely on the right track.
No, I think it's slightly simpler than that,
Oh,
actually.
okay, all right.
Yeah, yeah. I mean, on the outside, when you have extracted the T cells, you're not actually challenging them to tumor cells anymore. You're simply just introducing one protein. I think so. That's the simplest thing that you could envision is that, let's say you actually do biopsy of the tumor sample. So you know there are certain markers, proteins on the tumor that are expressed more than your normal cells, for example, from anywhere else in your body. And you just pick that one target. That's not
Mm,
the best way to like be super um, precise or competitive, but I think so. It is the first pass, and it has done the job. So you just introduce this one protein that can bind this thing that's on the cancer, and so that it just goes and
yeah,
binds it, and then
yeah.
it does its recognition. But what you're suggesting is actually more, I would say that that is the way the therapy has to go. I think so that it should have this potential that it's a whole suite of cells, right? Like that is good to defend against many mutations, so that anything that develops more cancer inside is also tackled, such that you don't have a reoccurrence of the cancer. So, I mean, on the training side, I think so. There's quite some underexplored things that one could
Yeah.
envision and try. Yeah, but that's a very active and kind of upcoming thing, I think.
All right. Okay.
Yeah,
So more research proposals to be written then. Yeah.
yeah,
Yeah. All right. So I do realize that you know we've sort of overshot our time together. So I'll try to wrap it up a bit more quickly. Um, so well, it's great that we did cover all of the things that we listed about your interests before. Um, so would you like to talk about research ideas or initial interests, initial sparks that sparked interest in your current research interests, and through which you sort of gained a passion to pursue it as a career?
yeah. Yeah, I think so. It is kind of nice to look back and uh, at least to talk to a younger version of myself and see how it has been. I think so I really like telling and talking about that journey. It is a special one, and I yeah, I would like to kind of mention that yeah, I mean, I for me it was really important to start listening to what it is that really interests me and not doubting myself too much about even uncertainties at points of time where decisions did not seem clear. Um, yeah, I always had this idea that, people in a spark of a moment just realize what they are going to do for the rest of their lives. Um, I am, yeah, I don't like to ascribe any moment in my life to like being so momentous in that same sense. I feel, I mean, there were certainly things that were going on inside me. I felt something about my experience. I had to live that experience. I had to learn something from it. And something in me processed all of that and told me something that had made a decision at that moment. And for me, I've just I would say, I wouldn't call it given up, but I trust my instincts to work with the way whenever they need to, and that allowing me to make decisions and feel I have never felt anything that I've taken a decision upon has been personally bad. So I think it's a fully functioning, capable system that
Yeah.
I should just trust more and go with it. And yeah, it doesn't need to be loud and like a very epoch defining thing all the time.
Yeah.
Yeah, yeah, every learning experience just adds on to a better decision, I think. So, yeah, I think so, being open to ideas, to exploring, to trying to question why is it like you really like something, going through the intricacies of the subject, doing it with your own hands, building a personal feel for the subject, what it is, what aspects of it you like. Um, I think so. Those are important questions. You need to kind of find answers for
Yeah.
yourself, and yeah, that helps. So, yeah, that is something that I've learned. Um, yeah, on the big idea side, I think uh, yeah, I think so. Every year, I think so bigger than the last year. Idea just occurs in my head. Things are moving fast. Like AI is moving fast. Things are changing, which means our world model has to adjust to that now. So I mean, in that sense, I think so. We discussed this. Like protein engineering as a field is a very is a good modular way to actually think about any kind of proteins. Really, that if you and believing in this aspect of being able to use you know deep learning tools for uh, learning things that are not super obvious to us like immediately. I think so. In biology, there's a serious lack of good curated data sets apart from the PDB that are standardized in ways that we can extract meaningful things. And I think so. A lot of like, I think so. We should game those data sets better in the community to make more people participate in it and make sense out of it. That is one thing that I would love to see more, and more of like automation to make things more standardized is also something I'm very excited about. Wish to see it happen. Um, and yeah, the other aspect probably is yeah, since I am working towards developing tools for neuroscience and making this whole brain activity measurements kind of a thing, hopefully in the near future, I do really wish to like see more um, kind of on the mechanistic sides of neuroscience things happen on brain-wide scales, and I feel some of these tools will help unlock some of those kind of questions where we kind of start to understand what a brain computes like, what does it care about, what are some of the variables there. But yeah, that's kind of the top two, I
Right.
would say, in my mind.
Yeah.
Yeah.
So it's actually deeper than just neuroscience. It's more like bridging like building a more concrete bridge between the cognitive sciences and to just connect how the brain really works, not limited to neuroscience, but even beyond. Yeah. Right. So wrapping up, would you like to share like specific challenges in your research where you had to think creatively to overcome some, that specific challenge, and what was a failure in that experience that taught you valuable lessons? And it doesn't necessarily have to be like a material failure. It could just be frustration with a particular train of thought that you think that you're really interested in this one particular thing, but you dig deeper into it and you find out, okay, it's not all that you imagined.
Yeah.
So, for example, your
Yeah.
transition from, what I'm referring to.
Yeah.
Yeah.
Yeah, I think so I should mention that. I think so when I was doing, when I was part of more computational biology projects, I think so there is certainly a satisfaction that comes with having numbers in your hands and statistics in your hands that you can make meaning out of. Um, although it kind of hit me hard really, that that I felt actually it was quite unlike almost anyone else near me at that moment, that I felt this bizarre gap between trying to be a bridge between, inference-based statistics from being a computational biologist to actually being able to be a biologist who wanted to test hypothesis one could build from such inferences. And I felt like everything was very piecemeal. Like the way even I learned biology is very piecemeal, in the sense that there are corners of biology people go very much into the depth into. They're married to their tools, their ideologies. They don't try to bridge it with something on the competition side or on another scale of those same things. And I felt like I was not married to any kind of like any of those corners really. I wanted something that bridges everything. Uh, but yeah, I felt quite starkly that at the end of my master's, I really wanted to do a hard switch to doing more wet lab biology. I really started with any project anyone would give me in a wet lab, and that turned out to be protein engineering, and that's how I started in the field. And it led to one thing and another, but um, to me, I think so. It was frustrating to see that as computational biologists, at least at the moment, people were not thinking about testable hypothesis generation in the biological realm at all. Like you could build tables of statistics and not be able to verify any of those. And I did not personally see a lot of meaning as a biologist in it. If your inference ends with an Excel sheet, I wanted to go into iterations with experiments and come back and refine the hypothesis and do the whole spiel of like, the scientific method. That's what I don't know. I signed up for, in a way. So, yeah, that's how I felt. And since then, I think so. I've been doing more wet lab biology, tool building, and hopefully the motivation there being that some of these tools will actually help to generate more richer data sets, and maybe it all comes back full circle, and I can be like a fully capable biologist who does both computation and wet lab stuff. And yeah, something of that sort.
So like a very holistic biologist who sort of just bridged everything. Then that makes sense because I think biology that way, because of the scale at which the complexities exist that we have discussed throughout, that's been a central theme of this episode since the beginning. I think it's been
Yeah.
very hard to build proper theoretical, build a proper theoretical framework that sort of unifies, multiple scales of complexity, and you're just able to sort of build a computational model out of that theoretical framework. And I think that's what you're trying to do here, is to actually deal with data in a way that you're able to either repudiate old theories and build, not either, you're able to repudiate old theories of dealing with computational data, or basically a theoretical framework, and build new ones that probably are more holistic in nature. Did I get it right?
Yeah, yeah, that's
That's
the
more or
hope at
less.
least.
Yeah,
Yeah.
great. So last question is, this is more of a get to know you question. What were your favorite and least liked subjects in school, in college, which I suppose you've answered, so you can pivot more towards school and right now. But you definitely haven't answered the least liked subjects part for college. And
Yeah.
following up on that, what's an inspirational thing a mentor or a teacher has said to you that has inspired you in your research philosophy?
Yeah. Um, I think so. Going back to school, maybe I really enjoyed maths and physics, I think. Chemistry a little less, but that was because I don't know, I didn't really like inorganic too much. Later on, I started liking organic and physical, so I cannot even say I totally dislike chemistry. Um, and yeah, physical being almost like physics to me. Uh, Biology, I never thought too much about it, but my impression, at least learning uh, all those phylogenies, was that it was very boring, and then I didn't have any interaction with biology whatsoever. I don't know. I was a bit. I want to say I'm a very excitable person. Like I really liked every subject. Uh, I really liked English literature. I write poems, so I still, yeah, I enjoy that. Um, yeah, maybe biology was the most least interacted with and had this dogmatic feeling that it's really about mugging up. Probably
Yeah.
that's the most boring, according to me, then, back then. Um, and inspiration wise, I think, yeah, I feel like I grew up in an environment which was very, I don't know, kind of research supportive in a way. My father is a research scientist, so mechanical engineer, and I have like we have studied a lot of physics together. Like he has taught me a lot of maths and physics, and kind of biology is a subject that I picked up on later. So now he tries to like play catch up, for trying to like find new things and tell me about it and ask me about it, which is kind of funny. But I think so. Yeah, I think so. My father has been a big inspiration in the sense that um, his curiosity is something that I've noticed since I was very young. Like any new subject, any new book he would see in the library, he would just get like printouts of it. Um, and he like tried to like engage with the subject very systematically. He questioned everything on paper, like things of that kind of at least uh, can at least instilled in me a feeling that I don't have to take anything on paper to be for granted. Like I
Yeah.
need to question this and where is it coming from, and have to develop my own feeling for it. So that is something that kind of just has been there with me. And yeah, I think so. He is like a super curious individual. Like uh, talking to him is just like going down rabbit holes. So I think so. personally, then I just became attached to rabbit holes, I think. Yeah, so it's like
Yeah.
chasing one rabbit hole after another. So, yeah.
So Alice
So
in
that has been,
Wonderland then.
yeah, kind of like that.
Yeah.
Yeah.
All right.
So,
Well, thank you so much for coming on this podcast. It was
yeah,
really
thank you so
nice.
much for having me.
Thank you. I definitely hope to have more interesting conversations sometime down the line with you. We can probably pick something and. But thank you. I really appreciate it. I learned a lot, many things, and I hope that the audience did as well. Yeah. So I'll stop the recording now. So thank you once again.
Yeah, thank
All right.
you. Okay.